Thank you very much, everybody, for coming along tonight. I'm Pete Boardman, and I'm going to talk about craneflies, a little bit about their ecology, their identification, and really their all-round wonderfulness. And I hope you'll enjoy the talk. So tonight we're going to talk about my journey into cranefly recording and taxonomy. So I'm going to be a little bit self-indulgent, but I hope you'll enjoy it. We'll talk about the relevance of craneflies, why they're important. We'll look at some interesting cranefly names because they're called all sorts of different things around the world, but not only around the world, around the UK as well. We'll look at where to find craneflies. We'll look at cranefly families and how to identify them. And we'll have a little foray into the history of British craneflies. The new book, which was out very, very recently. It's been a bit of a frustrating journey, but ultimately a rewarding one. And then we'll finish with a few legendary names and cranefly recording. So on we go, and let's hope this works. So craneflies for me started on a very precise date, the 10th of May 2001. And I know that because the photograph on the right-hand side was one I took on that date. As the image suggests, it was taken on an old photographic slide. None of this instant gratification of modern-day digital photography. I saw this thing in the field and crept up to it, took some photographs of it, and then I used the rest of the film up, and then I loaded it from my camera and put it in an envelope, and I took it to the post box, and I waited about a week, and then it came back, and then these images emerged. At the time, I had a suspicion what this species was, but I didn't really know anything about craneflies. I'd not looked at them before, and I had a feeling it was quite an uncommon species because it had been described to me by somebody we'll encounter a little bit later on in the talk, called John Kramer. He'd been doing some work at Fens and Wixel Mosses in Shropshire, where he'd been assessing the cranefly list, and he'd put an outline description of this pretty little cranefly with striped wings and an orange abdomen. And so when I saw this, I thought, oh, that could be quite exciting, because at the time it was known to be a Red Data Book 1 species. I then sent a printed version of the photograph, again by post, it was before the internet, to Alan Stubbs, who again is somebody else we'll meet later on in the journey. And he phoned me back. Again, this is before email. He telephoned me and confirmed that it was indeed this particularly rare species, and encouraged me to do more observations on craneflies. And so that was my sort of journey in. Along the way, I was helped by Tom Maudsley on the left from Liverpool Museum. He was the curator of Diptera. At the time, I was a volunteer and encouraged me greatly. And secondly, Alan Stubbs on the right-hand side was very, very encouraging and supportive. And so suddenly, craneflies were my thing. Before that, I'd been really into moths. I was a Shropshire moth recorder for a while. And so I was used to craneflies coming into the moth trap, but had no real understanding of them and didn't know anything about them at all. But now seemingly I was hooked. Then in 2003, I started to take craneflies a little bit more seriously and started to write a few brief scientific papers with observations, initially finding Idioptera linei, the fly in the photograph previously, is new to Wales in 2003. And then I started to do a little bit of research and try to find the larvae of the species, encouraged by Alan again. I was successful, and I was able to publish a little article demonstrating the larvae and the pupa of Idioptera linei. Previously, it was unknown to science. So again, it shows you how, even as a beginner, you can make really interesting discoveries with a little bit of persistence and a little bit of guidance and support from others. So the Idioptera craneflies were the first craneflies that I was able to research, and I carried that forward into an MSc dissertation eventually. I did a bit of work comparing the habitats of both species of Idioptera that we have in the UK, Idioptera linei, Idioptera pulchella. The difference between the two is in the females, the major difference. The females of pulchella aren't capable of flight. You can see there in the photograph they have reduced wings, so they're unable to fly, whereas the females of Idioptera linei can fly. That's what the larva looked like in amongst sphagnum moss, and it was collected from a number of places in that sort of edge of open water zone where the sphagnum moss starts to really dominate Sphagnum cuspidatum. I found it on a few locations at Wixell Moss and at another site. And I published my dissertation in 2005. Probably the most interesting thing I found was that if you look at all the records of Idioptera linei, the rarest species, and plot them into a phenology histogram, it shows that they have a sort of a roughly single generation phenology. But if you split them up with the northern records from Shropshire northwards and then Shropshire southwards and split them and plot them, you start to see the difference, and you get the northern population having one generation with a peak in June, whereas the southern populations, or relatively southern populations, have two generations with a peak in late April to early May, and then another one in late July. So that was probably the most interesting thing that came out of my my dissertation. It was good fun to find out. As part of my field work for my dissertation, I travelled all over the country, down into the New Forest and up into the Kielder Mires, and really got to grips with craneflies. Spent a lot of time recording. Really interesting way of getting into a group, going to lots of different sites in different parts of the country, seeing what's there, collecting them and identifying them. And it was a really good grounding. By that time, I was completely hooked by craneflies. So the next logical step for me was in 2006 when I started to work on a cranefly atlas for Shropshire. It covered eventually, on its publication in 2007, 233 species based on about 4,200 records. And the original idea was that this book would be published to coincide with the publication of British craneflies, more of which later. British craneflies didn't appear in 2007, as you all probably know now. But my book was out there. It gave some very basic information on species. It mapped all the locations in Shropshire where they occurred and gave some sort of ecological information about the sites on which craneflies occurred. In 2008, something interesting happened. I was in work one day and somebody came in to see me with a load of tubes that had come from Cameroon, and they were craneflies that had been salvaged as bycatch from a couple of Malays traps that were run on the side of Mount Coupe. And the person who brought them in gave me them. He knew that I was into craneflies, and he said, these are for your retirement. I subsequently put them straight in a drawer to forget about them because I had no idea how to go about identifying African species. I'd only just produced a local atlas. I was by no means an expert on UK species even yet. So I think this was a little bit out of my reach at the time. Here's a representation of the drawer into which I put the craneflies. I would note that this isn't the real drawer that the craneflies lived in, but merely a representation of a similar drawer in which they entered in 2008, and they emerged a number of years later. This was the year in which they emerged, 2014. I was lucky enough to get to go to the International Congress of Dipterology in Potsdam in Germany. At the time, I worked for the Field Studies Council, and they very generously allowed me to go through my Venn work project. The tagline of the work project was Shropshire and the wider region. And so we made the conclusion, probably the sensible conclusion, that the wider region included Potsdam in Germany. And so that enabled me to go. I spent a bit of time there with Erica McAllister, who many of you will know. Erica is an incredible advocate for flies, and she introduced me to all the world experts that were at the conference. And sitting through Erica's talks and also a variety of other talks from people around the world, it sort of fired me up, and it elevated me from what I considered myself to be at the time, which was very much a parochial dipterist who was only concerned with flies in my particular area, to potentially one who could take on the world. And I really would recommend, if anyone ever gets the opportunity to go to one of these conferences, it's incredibly inspiring. So the next step in my journey was to revisit the Shropshire Cranefly Atlas. British craneflies still hadn't appeared by 2015 when I started work on this. And so I embarked in 2015 on a lot of field work around Shropshire. So 4,200 records became 10,000 records. The taxa, the number of species in the book, went up to 245. But the main difference between this book and the previous book was that rudimentary keys appeared to the Shropshire species. And we felt that was a good move with British craneflies not yet out. And the available keys that people were using were old test keys that had often been photocopied and photocopied again, and were becoming more and more unclear. So the idea was this would, again, act as a bit of a gateway to people who were interested in craneflies, but who weren't able to successfully use the keys without some further bits of help. So the book contained hints and tips and photographs, and was aimed at people taking an interest. For me, along my journey, again, the next stage, I was very fortunate to be awarded a Churchill Fellowship in 2018. This enabled me to spend a bit of time at the Smithsonian Institution in Washington, DC. I was there for about a month, and I was able to take those craneflies out of the drawer at the FSC and start to look at them. And the main reason for going to the Smithsonian was that it inherited Charles Alexander's vast archive of craneflies when he passed away. Alexander was responsible for the identification of 95% of the known species from Africa. So it was the place to go to look at his specimens and spend time comparing them to my specimens seeing if they were likely to be similar or different. And that was just an incredible experience. I came back from Washington and started work on the specimens, and then eventually in 2020, last year, I was able to publish a paper in Entomologist Monthly Magazine highlighting 21 new species of craneflies and a new fold-winged cranefly from Mount Coupé, Cameroon, along with notes on a further 18 species that were new to the country from the same location. So it was a bit of an incredible journey from that first arrival of the specimens to me to be able to publish. This is one of the species that I encountered in the collection of tubes. I named this one Tanyptera Zorro due to the Z shape. On the right-hand side, bottom of the wing there, you can see there's a black Z, just like the mark of Zorro. I'll make the noise for anyone who may remember it from their childhood. You can't see me, but I acted out the Z at the same time. So that was one of the more entertaining species that I came across. Following that, I was given some specimens from Corsica, and most recently identified one of those new to science with the help of Dr. Jaroslav Stary from the Czech Republic. So that's sort of my cranefly journey, really, from first finding an interesting species back in the days of slide photography through to where I currently am now. So we'll move on to craneflies. In the cranefly book, British Craneflies, there are a list of names of craneflies from around the world. These are all different to those lists, and I thought I'd include them because there's some really interesting names. On the UK Cranefly Recording Scheme Twitter page, we asked people from all around the world, what craneflies called, and here were some of the responses. I'm not going to attempt to pronounce any of them because I'm a coward, but you can read them. But I particularly like Big Alistair of the Ponds, the Gaelic name for craneflies. That's really, really very entertaining. I also thank Jorge, who's on the webinar tonight, for the Latin American name. I'm going to try Zancudos, is how that's maybe pronounced, probably not though. I also like the Maori Peki Peki Haratua, hopping things of the May season, which is entertaining. So clearly craneflies have earned a place in the culture of many civilizations around the world. So what are craneflies? Well, they're true flies. So all true flies have a single pair of wings and halteres, and they sit within the suborder Nematocera, belonging to the superorder Tipuloidea. There are around 16,000 recognized species worldwide, and that's probably a massive underestimate. There are lots and lots of new species being recognized every year, even in this country. So currently we have 340 known from the UK, and they represent 4 different families. The craneflies, the Nematocera, sit within that suborder known as the threadhorns, and they represent the groups of flies, including St. Mark's flies and fungus gnats and owl midges, winter gnats, window flies, fold-winged craneflies, mosquitoes, black flies, etc. I've shaded out the other 3 families because clearly they're of less importance and all are less wonderful than craneflies. This is the good thing about Zoom. Nobody can take me down. I'm in control tonight. Cranefly larvae are really entertaining things. They have some wonderful forms. So each of these photographs, we're looking at the the anal plate, and the two things that look like eyes on each of the images are spiracles. So that's how the larvae breathe. And then below the spiracles, there's an anal opening. And the sort of endless combination of these features, I find really entertaining. You can easily while away an hour or so on the internet looking at the anal plates of craneflies. I also invite you to do that. There's one massive myth about craneflies that we have to get out of the way now, and that is that it is said that daddy longlegs are amongst the most venomous creatures on earth. They lack any means to inject their poison into humans, which is absolute rubbish, as I'm sure you would recognise. So we'll look at the relevance of craneflies now. They have a very, 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 very minor role in pollination, but I think it should be mentioned that they do have a role. Generally, most craneflies don't eat, they don't feed as adults, and any pollination that's done is accidental, apart from a few families, a few groups. Most of them are accidental pollinators. They do, however, have a major role in the food chain. Lots of things eat craneflies, and if they were that full of venom, nothing would touch them. They are known to improve the soil structure, particularly larval stages of soil-dwelling species. There are well-known pest species, the Tipula Paleodosa, Tipula Oloracia, etc., of man-made grassland, particularly lawns and amenity grassland. But they may well help the balance between grasses and herb species in more natural habitats. In forest soils, they have a major role in litter decomposition, and decomposition happens twice as fast where tipulids are present. So you really could call them ecosystem engineers, and I think that's a term I'm going to start using. When people complain of craneflies as pests, I'm going to counter with the ecosystem engineer argument. And again, I ask you to join me with that. Craneflies need all the advocates they can get. Some of our most threatened bat species, horseshoe bats in particular, feed on them in pretty good numbers, as do some of the smaller bats. The pipistrelles feed on some of the smaller species. In a really interesting recent DNA analysis of food sources for Bechstein's bats in Oxfordshire and Buckinghamshire, two species of cranefly, or the remains of two species of cranefly, had been found as part of a single meal from testing Bechstein bat dung. There was a species of Nephrotoma, so quite a large cranefly, and a species of Mellifluous, so quite a small cranefly, but interestingly, part of the same meal. So craneflies clearly are an important food source for bats. They're around pretty much most of the year, in all but the very, very hardest winters. You can see craneflies flying around. I've seen one on Christmas Day, on a mild day, but they have a couple of peaks. They sort of peak up into the early summer, and then they sort of maintain species numbers, start to fall again, but then that levels off in autumn when the autumnal species come out, and then craneflies can be found well into November, to sort of 15 species or so. I recorded here in 2012 as part of the field work for my second book. That makes them a good target for entomologists, because most dipterists hang up their net from sort of October onwards usually. So there are craneflies out in October, there are craneflies starting to emerge in March. So they're a good target to study. No, they're not. How do you recognize craneflies? All craneflies have a transverse suture, which is this sort of V-shape mark across the middle of the thorax. It's more obvious in the larger tipular species than it is in the smaller ones, but it's still there. So that's how you tell you've got a cranefly. But in terms of identification, the wings and wing venation is really, really important. And often when we do training events, one of the first things that we show people is where particular cells are that are really, really important in using the keys. The discal cell, for instance, in this case, this is a closed discal cell. It's a cell that is surrounded on all sides by veins. And we compare that with an open discal cell on the top image there. It isn't surrounded on all sides, it's open through to the edge of the wing, so we would class that as an open discal cell, and that's really important in keying out those particular individuals. As well as the veins, coloration can be really important in terms of identification. Some species, as you saw in the very first slide of craneflies that I showed, have striped wings, well-marked wings. Some have lunules that run either part way or full way across the wing. Also on those two photographs, you'll see black areas to the top right of the wing. That's the stigma, and the coloration of the stigma can be important. It can be clear, it can be brown, it can be black. So that's often mentioned in the keys. The heads of craneflies are important potentially. That could be the shape of the head. The picture top right is one of my Cameroon species with those two tubercles that are really prominent, which is quite an unusual find, I think, but not unique. On the left-hand side, the two photographs there, the antennae are really important and will help you to get towards certainly a family, if not a species in some cases. And then the bottom right picture, the rostrum and the sort of nose of the cranefly is extended in some species and really, really extended in others. This is one of the Elephantomya species, and you can see that the length of the rostrum there is sort of roughly similar to the length of the femora of the leg. So really, really long, almost as long as the wing, in fact. Also, in terms of identification, the thorax is important. It could be coloured in a particular way. It could have markings in a particular way. It could have hairs, particularly either on the scutellum or the postnotum. The pleura, the side of the thorax, is often important in terms of identification, again down to colour. Sometimes the top sections of the leg, the coxa, are important. They could be coloured in a particular way. So it's a bit of a mix and match, really, as how to identify craneflies. It could be important to know the coloration or patterning on the tergites. It could be important to know the shape at the end of tergite 9. Likewise, on the underside of the cranefly, the sternites, there's often a shape in sternite 8 in the tipulids. The outer clasper there, the shape of the outer clasper part of the genitalia is important for most species, particularly obviously the males we're talking about here. And in terms of the genitalia for male craneflies, sometimes the shape of the inner clasper is important as well. And to actually see the inner clasper, you have to remove the outer clasper. So a little bit of dissection is needed, but generally not too much. And I sort of wouldn't compare the level of dissection needed to some other groups of invertebrates. They're generally reasonably okay to do. In some of the other families, these are both short palped craneflies. You can see the styles. That the specimen on the left-hand side are really long and sword-like. On the right-hand side, the main style almost has the shape of an elephant's head with a bit of an ear and then a trunk. So they come in all sorts of shapes. Generally, it's comparing shapes with images that are either in the key or elsewhere. So we'll look at the 4 families now. The first family we'll discuss is the Cylindratomids, the damsel craneflies. As the name suggests, that they are very, very narrow abdomen. You can see the photograph on the left there. The abdomen, particularly the first few segments, is barely bigger than the legs, and you instantly get to recognize them when you're used to looking at the larger tipular craneflies. All of a sudden this is different, and it appears to be very, very long and slim. That's the Cylindratoma species, which is a sort of woodland edge species principally. On the right hand side is the larva of one of the damsel craneflies. This is actually Triogma trisulcata. They have more of a caterpillar appearance than a sort of a grub appearance, and they're the only family of craneflies who have this sort of caterpillar appearance to them. They principally feed on bryophytes. In this case, Triogma feeds on bryophytes in fen and in some cases bog as well. The next family is the hairy-eyed craneflies. There are 20 UK species. The actual hairy eyes are often quite difficult to see, even under magnification, unless you get the lighting right. And here you can see that the light has been angled in a certain way that shows the hairs as white, but believe me, they really don't stand out like that normally unless you get your lighting correct. You can just about see them with a hand lens in the field, if you are lucky and can angle them in a correct manner, but generally it's a microscope job unless you recognize the individuals. For instance, the cranefly on the left, Pedicia rivosa, has really sort of telltale markings and can't really be mistaken for much else in the UK. The hairy-eyed craneflies split into 2 subfamilies. One section are aquatic and semi-aquatic in terms of their larvae, and the other group, the Eulinae, are fungi feeders as larvae. Probably the most familiar to most people are the long-palped craneflies, Tipulidae. We have 87 species in this country, and as the name suggests, if you look at the photograph on the right, they're named after those long palps, which in this particular case of this individual are about as long as the antennae. It's something you very quickly get to recognize when you're comparing long palped to short palped. On the left is one of our common spring craneflies in the UK. It's Tipula venalis. It's relatively easily identified in the field. It has slightly tinged markings on the wings, bits of white. It has an orange abdomen with a dark stripe down the middle, And in life, it most often has bright, shining green eyes, which you can possibly see from that image. But it's associated with dry grassland. So ideally, you'll find it on road verges, calcareous grassland, all those sorts of dry, grassy places in spring. The Tipulidae split into 3 subfamilies. The comb horns on the left-hand side, which are deadwood feeders, saproxylic species. The Tipulinae, they feed as larvae on a variety of plants and mosses, plants that are decomposing, and bryophytes, and also other situations. And then the white-footed ghost on the right is our only UK example of the Dolicha pizzinae, and they feed upon moss. If you can see in the photograph, you can see the white tarsi at the sort of top middle of the photograph, and they're a wonderful creature. I've seen them inhabit very, very shady, overhung trees on bogs where root plates have lifted up, and when I've disturbed them, they've more or less sort of flown at me with their legs spread, and all you see is this white on the tarsi. You don't see the black body. They're a wonderful, wonderful creature. I like the white-footed ghost an awful lot. The short pout craneflies, 229 UK species. On the left-hand side, a pretty common one that lots of people see, Limonia nubeculosa, with those striped legs and spotty wings. On the right-hand side is a species called Rapidia, Rapidia maculata. It has pectinate antennae, but you can see that the palps are really short when compared to the long palp craneflies. Likewise, with the tipulids, they split into 3 subfamilies: the spurless craneflies, the long-bodied craneflies, and the simple-veined craneflies. I've always been a little bit suspect about the use of naming something after something it doesn't have. I always thought spurless was an interesting bit of terminology, but it's being compared to long-bodied craneflies which do have spurs. So maybe it works. On the left-hand side is a mellifluous species. They have very, very hairy wings. In the middle is a Philodoria species associated with wetlands. And on the right-hand side, a Limonia species, which is a woodland species. In terms of size, our largest UK species is Tipula maxima. It's a wing length of 25 millimeters. Very, very spectacular thing. Always surprises people when they see it for the first time. Those wings are absolutely brilliant in terms of camouflage. It's a species that will fly around, and if it gets disturbed, it will head to the ground, and you do well to see it against the vegetation. Very, very well camouflaged. You'll find it in wet meadows. You'll find it next to wet woodland streams in those sorts of habitats. Our smallest UK species or group of species is the genus Tasiacera. This is Tasiacera marina, which is the commonest of the genus. It has a wing length of 3 millimeters, and you can see there 3 specimens compared to a 5p coin, which probably won't mean a great deal for some of our overseas viewers today, but pretty small things. And often they are amongst the remnants of craneflies and other insects crawling around the bottom of the net. They sort of rarely fly out, and almost impossible to see if you have a black net. It's where using a white net is really useful. Here we go again. We see these sorts of things in the papers annually. I haven't yet been contacted this year. I often am And in the past, I've tried to comment and suggest that 200 billion daddy long legs aren't braced for an onslaught, and people don't need to worry about them. But they appear in the newspapers anyway, irrespective of what you say. And I think I'm probably going to stop commenting on them because they're going to print what they want to print anyway. Craneflies first emerged a long time ago, about 230 million years ago in the Triassic. The hairy-eyed craneflies were the oldest of the four families. Then the short pout, and the long pout, and most recently the damsel craneflies about 50 million years ago. So they've been around an awful long time. And if you look at the maps on the right-hand side, you can see that the Earth has undertook many, many changes whilst craneflies have been around. And that sort of helps us understand the distribution of some species. For instance, this species here is called Symplectopilipes, and it occurs in all those countries around the world. It first emerged in the Jurassic period, or something like it very much did. And you can see with continental drift having not taken place in terms of how we would recognize our current alignment of continents, that probably helps to explain why the craneflies are all over the world, particularly that species. You can readily come across fossil craneflies on the internet, particularly from samples in China and the USA. This particular one is from the Lower Cretaceous period, around about 125 million years ago, in shale. There's a large body of craneflies that have been found in amber, and there are a few cranefly taxonomists who just specialize in species that are found in amber, probably from 60 to 60 million years ago or something like that. So where do you find craneflies? I've listed a few locations there that you could target to find things. But ultimately, you'll find craneflies in most habitats, particularly in the UK, certainly. And we'll go through some of those shortly. It's said you're never a metre away from a rat in the UK. You're probably not more than a few metres away from a cranefly in the appropriate season. And in the UK currently, we're just starting to see the emergence of Tipula paleodosa, the commonest species we have in this country, and they're all over the place at the moment. One came into the house I was in last night and flew around the lights, which was entertaining. I managed to catch it and release it. I like this photograph. This sort of illustrates the difference between what we could call old-fashioned farmland in the foreground and very much improved farmland in the distance. And you can see in the foreground there are all sorts of little holes that have water in them, very, very soggy bit of land that it's part of a floodplain of a small river. You can see the vegetation, there's juncus, and you can see it's tussocky and wet and absolutely wonderful for craneflies. Craneflies love that sort of habitat. The habitat in the distance on the dairy farm, you'll still get high numbers of Tipula paleodosa coming from that grassland and a few other species, but nowhere near as much as the grassland in the foreground. Lots of different types of wetlands are important for craneflies. We saw with Idioptera linei at the beginning of the talk. Sites with sphagnum moss are important for that species and a whole range of other sphagnum specialists. Swampy, wet areas like the grassland on the right with a bit of reed bed again important for a different range of species. This is some wetland, sphagnum wetland in the uplands, which has a sort of distinct population of a variety of species. And again, another wetland site, a river through a valley. This is slightly upland Shropshire. This is the Longmynd, for those of you who know it. And actually, you have a few sort of distinct habitats here that different species will be found in. So the river and particularly the river a little bit further upstream, where it's a little deeper, will have a certain set of hairy-eyed craneflies associated with it. Where the chaps are standing in the photograph there, it's quite close to some seepages, and again you'll get distinct species associated with that sort of habitat. Another type of wetland is tufa springs, and you have a whole set of species associated with the seepages around those and the wet areas around them. Plus you have one very specialized species that's associated with bryophytes that grow on the tufa itself. And you can see that tufa spring is within a wet woodland. Really wonderful site for craneflies. Anywhere that you get accumulations of wood in river systems is important. You get a whole raft of species that will come out of sodden dead wood, or dead wood that's a little bit drier at the side of streams. So very, very rich habitats. One of the sort of Shropshire specialities is the Dingle Woodland, where you have quite a narrow stream through a river valley, or through a valley. And again, really, really good sites for a whole range of things, including Lipsothrix craneflies, which are a saproxylic species. Here you can see a couple of examples of a larval exuviae with one that's hatched and left it behind, coming out of some wet sodden wood. And on the right hand side, a teneral Lipsothrix that's just emerged out of the dead wood, and that will be flying away relatively shortly. There are a set of species associated with river shingles at the sides of rivers. A couple of Eleophylla species here, Eleophylla apicata, very well marked wings, which readily identify it. Likewise with Eleophylla virale, the spots on the wings there readily identify it from the habitat, that sort of habitat. They're both nationally scarce species, but are readily found in the right sort of habitat. There's a whole range of calcareous loving species. I won't go through that list, but so it can be quite important to understand the geology on which you're looking for craneflies. Likewise, there's a reasonable list of things associated with acidic habitats, including what we can consider now to be our old friend Idioptera linnaei. Craneflies do turn up in odd places. This photograph was one that I took when I was in Queensland in Australia. I was walking through an area of mangroves towards the beach, and my eye was drawn to a spider's web that had a number of craneflies sat on it. And I took a photograph, which is this one, and then I disturbed the web to see what would happen in case these things were actually stuck. But no, they flew away, or they flew, and then they came back to the web and took up this formation. So they occur in all sorts of situations. It's very unlikely that much has been done about the ecology of this species. It was one that Charles Alexander named based on specimens that were sent to him. He never visited Australia. He only identified stuff that people sent to him from around the world. And that is one of those species. I'm going to talk about the Cranefly recording scheme a little bit now. So it was founded by Alan Stubbs, who we've seen previously, in 1973. Alan was assisted from about the early 1990s by John Kramer in the centre, who still publishes the newsletter. And I got involved in about the 2010s, starting to support Alan and John. I took over as recorder from John very recently, about two and a half years ago, and Alan's sort of responsibility has been writing British Craneflies, which we've talked about and we'll see again shortly. Bit of a quiz question for you. What do all these people have in common with the Cranefly recording scheme? They are Professor Alice Roberts on the left-hand side, Adrian Brody, the actor, and Noel Fielding below, Kate Beckinsale and Pharrell Williams, the singer. And the answer is they'll all be 50 in 2023 at the same time as the Cranefly recording scheme. Wouldn't it be amazing to get one of those good folk involved and celebrate with us? In terms of the scheme, we're always looking for people who can help. Currently, there's just Alan, John, and myself, and we have a little bit of help from a couple of other people on Facebook who run our Facebook page. But with the release of British Craneflies and the anticipated uptake of the group by more people, recording is going to become more onerous. And so if you feel you could help in any way, please do get in touch with me. So what we're planning to do is run a number of face-to-face Cranefly identification courses around the UK next year and the year after. These are at an early stage of arranging, but I've sounded out a number of people on Twitter via the UK scheme, and people have suggested potentially holding events in the parts of the country listed there. Again, could you help in arranging a Cranefly event? Do you know of a venue or groups of willing attendees? Again, please do get in touch with me, either via Twitter or directly. If you want to know more about the Cranefly recording scheme, we have a website we're sort of hosted as part of the Dipterus forum. There you can download recording scheme newsletters. You can see the original one there in 1973 that was released, and then the more recent ones. Often there's 2 a year, depending on the level of submissions. And you can see there the Cranefly recording scheme Twitter site. We currently have around about 1,730 odd followers. I'm sure we'd welcome an awful lot more, and we do follow back. Please give us a follow. So one of the things that we do in the recording scheme is to produce distribution maps. The map on the left-hand side is a list of all the records we have. So by hectad, so by 10 kilometre by 10 kilometres square. And you can see that the best recorded squares have up to 158 species recorded from them. But there are many, many squares that only have less than 20 records. So we always welcome records from wherever you are in the country of whatever species you have. These enable us to give really good population maps. The map on the right-hand side is a comparison between Cranefly recording over 2 distinct periods of time, and they give us a really good idea of what's going on with these species, and whether we can see any increases or declines. Along the same scope of that, the targeting revisiting maps, which UKCEH have enabled us to use. These work on the basis of the pink dots on there are squares that UKCEH would like people to revisit to see if they have the same species in them or more species than previously. One of the best things about that map for me is that Shropshire stands out very, very well as a result of the recording for the Shropshire Atlas. Imagine if every county had that level of recording, we'd know an awful lot about our craneflies. As I say, it works like this. So you click on a pink square, and that tells you the records, the species records that were previously recorded, in that case 2016, and it invites you to go back and re-look for those species, and the maps are updated daily with new species that come in. The other thing you can see about that map, there are lots of squares, lots of 1 kilometre squares, monads, that have no records at all whatsoever. This one is close to where I live, around the northwest of England, and as much as anything, that's a call for me to get out next year and do a bit more recording around where I live. Of course, having all that information enables us to do other things. The Cranefly Recording Scheme have been working on a review of the status of craneflies of Great Britain. It's been rumbling on for a little while, but hopefully it will be finished and published this coming winter. Another thing the recording scheme does is provide online little guides. This one is available for the tiger craneflies from the Dipteris forum site, and I've put links to it on our Twitter account on a number of occasions. Likewise with the Limonia species from Woodland Edge. Please do seek those out, download them, and have a look. Here we go again. This was a quote from, supposedly a quote from an entomologist in 2014 that appeared in a paper. It says, about craneflies, people will start to see them even more in the last couple of weeks. What even does that mean? As they head indoors for warmth, shelter, and to find a mate. Do they really? The craneflies can grow up to 4 inches in length and could be inside our homes laying eggs within days. Really? I don't think so. That's 4 inches, not against my hand. Have you ever seen a cranefly 4 inches long? No, neither have I. Okay, so British craneflies. I first came across it as a reference in a book as in prep in 1990. So it's been going for a long, long time. 2007, we were told it was definitely coming out in 2007, hence my Shropshire book, to give it a local context to the national work. By 2015, it still hadn't appeared, and I was persuaded to update my Shropshire book with identification keys. And finally, it was published in 2021. So British craneflies is actually the same age as these people, some of whom you may recognise. The Liverpool and England footballer Jordan Henderson, the actress Emma Watson, the singer Rita Ora, and then somebody called Joey Essex in the middle. He's the same age as British craneflies. Makes you proud. Very quickly, I'll look at just a couple of people who were fundamental in the development of the study of craneflies. I've already mentioned Charles Alexander, 1889 to 1981. Definitely the father of craneflies. He was a professor based in Massachusetts. He described over 11,000 new cranefly species, but never left North America. As I said earlier, people sent stuff to him, and this equates to 1 species per day over his entire career that he named. Absolutely incredible. Published over 1,000 papers. was very much a team with his wife, Mabel. It wasn't all him. Definitely his wife was involved in helping him, supporting him, transcribing some of the papers. As I said earlier, he named 95% of African crane flies, and on his death, his collection went to the Smithsonian Institution, where I was fortunate enough to be able to use that collection. This is him and his wife, Mabel, towards the end of both of their lives. Mabel died first, and when she died, Charles gave up crane flies. He couldn't face to work on them alone. Very quick skim through just a couple of other people. This is Frederick Wallace Edwards, F.W. Edwards, who worked at the Natural History Museum. He wrote a really important work on a whole range of crane flies, and published the first key to small short Pout crane flies in 1938, and did a lot of work, sent a lot of specimens to Alexander. Alan Brindle from Manchester Museum did a lot of work on crane fly larvae during the 1950s and 1960s, and he published more than 20 papers on larval morphology. A bit of a local hero was Cyril Pugh. He was from Shropshire, and he was the father of Shropshire dipterology. So when I came to do my crane fly atlases, I was basically following some of the, the work he'd started in the 1930s and 1940s. And finally, Piotr Oysterbroek from Holland, who is the author of the Catalogue of the Crane Flies of the World, which is the one-stop shop for information about crane flies from all countries. And those of us who are crane fly taxonomists depend on it for its supply of literature. Piotr is the person on the right-hand side holding his hands up. Anyway. And then very, very finally, drunk wasps, sex-mad spiders, now a plague of 200 billion daddy long legs is heading our way. First came drunk German wasps, really? Then sex-crazed spiders, and the actual quote is sex-crazed spiders the size of mice. Now 200 billion daddy long legs are set to invade our homes. No, they are not. And please sleep soundly. Thank you, everybody.
Thank you very much indeed, Pete. That was an excellent introduction to the group. I really enjoyed that, and the recording scheme as well. Yeah, it's great. You're going to be doing workshops around the country over the next couple of years, and it's also really pleased to hear that about the forthcoming review of British crane flies, because we really use lots of those sort of Natural England reviews to sort of prioritise our species surveys. They're really important to us. And obviously, congratulations to all involved with the book. You know, it's a great achievement that it's finally come out. But probably my favourite bit is always the crane fly anal plates and the faces on those. They're just absolutely brilliant.
Aren't they wonderful?
So thank you so much, and you're okay to stay for a few questions. I don't think we've got many at the moment, but
that's fine.
Thank you. So we had one first from Raman Sen. Hopefully I'm saying that right. Which species is most likely to be on the Peak District moors at this time of year? A lot of one. I don't know. Yeah,
there are. If you went on the Peak District now, there probably would be easily 20 or 30 species that you could find with a bit of persistence. You'll get some of the larger tipulids, Tipula Oloracia, particularly in the wetter areas. Paleodosa is likely to be around. Tipula helvola. I could just go naming crane flies, but I would say, as we don't have all night, probably 30 or 40 species. And if the person wants to follow that up, I'm happy to correspond with them and supply an educated guess or do some work with the database and let them know.
Yeah, thanks. Great. And is it going to be a point where they really, so most of them are sort of finished for their life cycle for the season as adults, and is it suddenly going to have a big drop off a cliff in terms of the numbers?
Yeah, when we get into sort of winter proper, that cuts species down, but it depends on the weather. If we get a really hard winter, most species are larvae underground by then and pretty much protected, and you won't see much in the way of craneflies. You still will see winter gnats, of course, which I didn't cover tonight, even though we do record them as part of the scheme in the same way we do with fold-wing craneflies. But I thought I'd keep just to the tipulids today.
Right, thanks, Pete. Okay, then the next question came from Andrew Charlton. Are identification features generally well preserved in dry, dead specimens, or do they need any special treatment for storage?
They are preserved. It depends, of course, how you preserve them. In dry specimens, you can have some problems with facial features collapsing a bit as they dry out. But generally, the majority of coloration and other patterns are observable from dry specimens. In wet specimens that you keep in alcohol, obviously you're going to lose some of the coloration, and you're going to lose anything that depends on that sort of difference in color or shading. But it's often easier to look at the genitalia in alcohol. So it depends what you have, really, and a good sort of combination of both is really useful. I tend to keep most of my specimens in alcohol for practical reasons. I just don't have the space to curate them here, but I do have a collection that I access at the FSC, and also I come into Liverpool Museum and use the wonderful collections there, which everybody should do.
Absolutely. And do the legs, I presume, are not used at all in identification features, because they're a nightmare, aren't they, with dry specimens?
In dry specimens, they very readily drop off. Alan always used to call them optional extras, which it has some truth in it, but legs can be really useful in fresh specimens to identify particularly the femora, whether the coloration on the femora is in a certain degree one color, in a certain degree another. Of course, we've got those species that have spurs on the legs that help us to define the subfamily. So legs, to a degree, are important, but generally other features, if you don't have legs on the specimen, will help you get there if you have a bit of experience.
Great, thanks. So the next question is from Steve Docker. Hi, Pete. Can you ID some species from photographs, and others? For others, how do you prepare a voucher?
Okay. Hi, Steve. Good to see you. I reckon you can probably identify about up to a third from photographs if you have good enough angles. Often where people fall down with photographs, they take one angle, and so you can't see all the features you need. You often need a couple of angles, so a top shot and a side shot, for instance, or you need a good view of the wing venation. So a number of species are doable from photographs, yes. What was the next part of the question? Was it do with preparation?
Yeah, how would you usually prepare a cranefly in terms of curation?
So what I tend to do, if I'm going to keep them dry, I card them. I glue one wing down so it's flat against the card, so you can see the venation, because often in some cases they can crinkle up and you really need to see that venation. And I glue the rest of the fly. So so half of it, so one side of it is glued to the card, and you can see the other side. And then depending what the genitalia is like, I will sometimes take that off and glue it separately or keep it separately. Sometimes you may need to put the genitalia, particularly if they're small species, into alcohol to give you a better look at the styles and the array of the styles and the beak and the spines and things like that. So it depends on species, but if you come along to our training workshops, then we'll demonstrate that.
Great, and I know some groups of Diptera, people sometimes they pin them into the side of the thorax, don't they? So would that, do people do that with craneflies as well sometimes?
Yeah, they, of course, that can affect some features becoming obscured. You know, you get a whacking great hole that a pin has made through the pleura when there may be a feature that's usable. But the same can be said by putting a pin through the top. It can obliterate whether it has a view of where it has a central stripe or not. Generally, the large craneflies like Tanyptera Paleodosa are big enough that with a small pin, they're not going to damage any features that you would want to look at. But with small specimens like the Tanyptera, for instance, you know, that would be fun to pin anyway. They're really tiny things, and you could risk not being able to identify them by indiscriminate pinning. It's the sort of thing that comes with practice, as anybody will know. If they study a group, they know the best way to look at it and the features they need and how to set them.
Yeah. Great, thanks. So come on a course, Steve. Okay, next question was from Sarah Hearn. How do you distinguish Tanyptera Paleodosa from Tanyptera Oloracia? Am I saying that right?
Yeah. So Tanyptera Oloracia is generally about first, so it flies from May. So anything that could be either of them that's flying in May will be Oloracia. But Oloracia does have a second generation and overlaps with Tanyptera Paleodosa, so both can be on the wing at the same time. If it's a female, it's relatively straightforward. You can do it from a photograph. The abdomen of Paleodosa is longer than the wings, whereas the wings of Oloracia is the same length as the abdomen of Oloracia. So you can tell females quite readily, males less so. You have to look at either the genitalia, compare the genitalia to the keys, or count the number of segments of the antennae. And one has 13 and one has 14. I'd be guessing off the top of my head to say which is which without looking at the key. The other way is to turn the insect upside down and look at the gap between the underside of the eyes, and one is wider than the other. So you can do some in the field if they're females pretty easily by the wing length compared to the abdomen length, but the rest of the features you need to compare under a microscope with decent magnification.
Right, great, thank you, Pete. Okay, a question from Linda Pryke. What's their dispersal ability like? Do they spread far from their emergent sites?
They can be, like any other small invertebrates, they can be carried on the wind very, very readily. I've had a couple of things turn up in my pretty urban garden. I use the word pretty and urban together. Pretty not meaning beautiful, pretty meaning averagely urban. And one of them is a species of peat bog. And of course, Warrington has its peat bogs, and you know, not too far from me, probably 3 or 4 miles away from me, there's a peat bog, so it's probably come from there. If you remember earlier, I mentioned that 2 species of cranefly turned up in the feces of a bat, Bechstein's bat. One of them was mellifluous, and the species involved again was a sphagnum related species, and there aren't many peat bogs or sphagnum areas in Buckinghamshire or Oxfordshire, so it's likely that that has travelled some distance before it was snaffled. You also get examples of craneflies turning up on oil rigs where they've been blown out by weather systems. So like a lot of other invertebrates, they can disperse quite readily. Most of them probably don't in any one year, and they'll be breeding pretty close to where they emerged. In if the habitat is suitable.
So like with moths, do you have a few regular migrants that turn up in numbers on the south coast every year, and less so as you go further north?
Not really in the same way. I suspect that does happen from time to time. New species do get found, but I don't think we have the same knowledge or the same network of, for instance, the way that moth trappers actively go out looking for migrants, and that's not the situation with us, but undoubtedly that does happen. It must.
Yeah. Okay, thank you. Question from Andrea. How many species are just in Scotland? So does that mean, I guess, exclusively in Scotland? Is there some Scottish specialities?
There absolutely are. Yeah, the sort of River Spey area is a hot spot, and I would be guessing. I can't tell you the precise number, but I'm going to say probably 10 to 15 species probably only occur in Scotland. Probably 10 species on reflection, but certainly those northern habitats, in fact, are our most recent addition to the UK fauna was a northern Scottish species, which is really a sort of Arctic arboreal species, was found in the very, very north of Scotland. So yeah, there certainly are Scottish species. One of the issues with a lot of Scottish craneflies is that there aren't as many recorders who are working on craneflies in Scotland as there are further south. So it can be, really revealing, spending a bit of time working up there. I was fortunate enough to go up to Shetland about a decade ago and do a bit of work up there. I found a couple of species that were new to Shetland. So yeah, more work is definitely needed.
Great. Do you get any that are at the tops of mountains?
You do, yeah. There's a couple of species that are restricted to those sort of alpine habitats. Only a couple of species, but they do occur from time to time, and very, very rarely get recorded because people generally don't go into those habitats with a net. Yeah. By the way, was that an invitation to go to Scotland to look for craneflies as part of that question? Because
out of the Tanyptera region, I'm afraid. I would love to. Great. Okay. Question from David Hill. How long do adult craneflies last?
Right. No, normally just a matter of days, maybe a week on average. Some will be longer. The craneflies that I reared as part of my my Idioptera studies, I actually drove them back to the peat bog after about they've been adults for about 2 days, and I drove them back. I thought it'd be nice if they spent the rest of their time in their actual own habitat rather than sort of put us pin through them. And yeah, so normally about a week or so is what you expect with a cranefly. Remember, like a number of moth species, craneflies don't feed as adults, so they only have enough energy to do what they need to do: find a mate, lay eggs, and then they die. So yeah, short period of time.
Right. Thank you. Okay. Question from Steve Lewis. I'm very interested in the ecosystem engineers aspect. In our current climate crisis, do you regard craneflies to be critical? And are they achieving things that other creatures are not?
Well, that's an interesting question. I must confess, I made up the words ecosystem engineers in relation to craneflies about 10 minutes before this talk. It suddenly occurred to me that it was a good thing to say. I was aware of some of the relationships craneflies do have with particularly, forest environments. And I thought, yeah, I think it's something I should start. Oh, I've just seen that. If you want to come back to Shetland, yeah. Thank you, Keith. I may well take you up on that. Thank you very much. Sorry, I've got rather excited and lost my thread. Yeah, so ecosystem engineers. I think anybody involved in invertebrate conservation and getting the message across that the value of invertebrates now is probably the best time ever to be doing that, because of the climate crisis, the dramatic reductions in invertebrate numbers. Anybody who's seen Bug Life's sort of a number plate splatter project this year will be aware of that, and it's something that we've been doing for a few years. So I think it's a really good opportunity to rather than, because everyone always says with craneflies, it's the place in the food chain which is their importance, and it absolutely is. But also they are ecosystem engineers, and we have scientific evidence of that. So let's go with that, as well as the importance of them in the food chain. And yeah, I think that would be quite interesting to do a bit more research on that, and see what what other researchers have published, and build up a bit of a more body of evidence to support that. But yeah, that's an interesting point, Steve. Something I'd like to spend a bit more time researching.
Yeah, thanks, Steve. Thank you, Pete. Okay, we're on to the last question out of the chat now, from Clive. I guess a binocular microscope is essential. Can you recommend one?
There are a number of manufacturers out there who are producing good microscopes. It sort of, as ever, depends on your budget with these things. It's like cars. You can drive an old banger that costs you 200 quid, or you can go and buy a Rolls Royce at £150,000. They both do the same thing. They get you from A to B. Often with microscopes, though, it's not the cost of the microscope, it's the lighting, which is really, really important. But I've used a number of microscopes over my time. When I was a beginner, I spent 20 quid on one, which at the time was still a little bit of money. And it did me perfectly well. It was a fixed times 10 and times 20, but it enabled me to identify my first craneflies. I was absolutely fine. I've now got a really nice Leica microscope that cost about £4,000. But I've used all sorts of things. Some of the cheaper bits of equipment that are really good. GT Vision sell a good range of microscopes. They're based in Norfolk, and Brunel sell a good range of microscopes. They're based down in Wiltshire.
Chippenham.
Yeah, Chippenham. And you can go down to Brunel and have a play before you buy. Those are going to be in the sort of range from £300 to £700, something like that. So yeah, it just depends what your budget is. All of those. Do you have any more you can add to that, Gary?
No. Yeah, I would say you don't need to spend that much money to get something that will do most things. I presumably times 40 or something will do you most of the craneflies in Britain.
Absolutely. Yeah.
Even genitalia, for the bigger ones.
Yeah. It depends how far you want to take it. So, for instance, if you're interested in getting really good images of cranefly genitalia for publications, then obviously you need stacking software and all that sort of stuff. I don't have that. I tend to go in and use the equipment at the Natural History Museum in the Sanger lab, use their kit. But yeah, my old 20 quid times 10 times 20 was a good start, but anything that goes up to about times 40 is really, really useful and pretty much do most things you want it to do. There, but a couple of the smaller things like Tassiocerea, you'll probably need a little bit more magnification. And if you a really cheap microscope, sometimes times 40 is the quality of the optics can be struggling on cheaper models, but generally you'll be absolutely fine. Start off with a cheap secondhand something, and if you think that that's for you, then invest in something. I've had people talk and say, oh no, I know microscope's too expensive, £700. No way I'm going to pay that. And then they go on holiday for £2,500 somewhere. So it's about priorities, really. And not everybody can afford lots of money. I couldn't when I started out. So yeah.
But yeah, I think £200 can get you a reasonable times 40 actually nowadays. But I would, if you're anywhere near Liverpool, we still can't welcome visitors behind the scenes at the moment. But when we can, I'm very happy to take you through a whole spectrum and range, all the way from, I suppose a starter microscope, which will do most things, all the way up to the top of the range. Happy to take you through, and you're very welcome to come in when we can welcome people back to use our imaging suite as well to get some very high quality stuff. Yeah,
fabulous.
That's what we're for, really. We're a public body that you pay for anyway through your taxes.
I would say again to anybody who has the opportunity to go to the museum when it does reopen, take advantage of it. The folk there are very, very friendly and great facilities. I learnt my trade there over a 10-year period or so, and I'm very, very grateful for the opportunity I was given to go into the museum.
Yeah, thank you, Pete. So we haven't got any more questions in the chat, but I suppose we'll draw it to a close. But if anyone would like to just unmute themselves and ask Pete a final question or something, go for it now. Otherwise, we'll okay, Ian Strang, yeah.
Hi, hi Pete. I'm sorry, I missed the first part of your book. Unfortunately, I'm up in the West Highlands and doing a lot of letting, so I'm having great fun using the new book, which is fantastic. And we'd certainly be interested in having you come up and do a workshop up this way if you'd be interested to do that. I'll contact you about that. But I want to ask you about larvae. I do a lot of kick sampling, and I've got a lot of larval samples. Is it possible to get into species?
It is in some cases.
Is it worthwhile?
Is it worthwhile? It is difficult. I won't pretend it's not. And Alan Brindle's keys aren't necessarily that well available. That is one of the areas, and I think Alan acknowledges it in the book, where there still needs to be a lot of work. Somebody needs to spend some time updating Alan Brindle's works and finding those species that we don't yet know anything about, and bringing it all together so that it becomes more possible to take the study of larvae to the next stage. And a lot of records I see for larvae aren't down to species; they're down to genera. So a lot of the hairy-eyed craneflies that turn up in Environment Agency samples, for instance, will be down as Pediceus or whatever. But generally, we don't get many records in. I've had very few people, in this country anyway, are working on larvae. It's definitely an area we could improve. And yes, I'd love to come up and do some training up there.
Great. I guess DNA barcoding can really help the study.
That's the next again. That's absolutely. That's the next great step. That there are still a lot of UK species that aren't appropriately barcoded yet. Something that I'm going to be involved with next year through work is to search for a number of species that aren't yet barcoded. Part of the Natural History Tree of Life, Natural History Museum Tree of Life project that I've spoken about before on these talks, and no doubt others will have as well. So yeah, the idea is that under the Tree of Life, all UK species are being sought out to be barcoded, and that would be really interesting to get fresh material for a lot of these species from appropriate locations that can be verified. So there is more work to do on that, but absolutely yes, it's going to be a really interesting area, and it's going to break a lot of the taxonomy, no doubt, as more species emerge.
Yeah,
presumably it wouldn't be easy to rear larvae from running water.
I know it's relatively easy to rear some of the soil living species, but the wetland species are probably a bit more of a challenge. As I say, earlier, I reared a bog species by making a little mini bog in a fish tank, and successfully managed to rear a couple of Idiopteralineae. But as for aquatic species, I don't know, to be honest. I don't know whether anybody's read any out, whether anybody else on the call knows.
So yeah, big area for development there. This is from Matt. As a professional taxonomist in the US, I'm concerned that DNA barcoding and environmental DNA analysis will be the end of professional taxonomists like us. I appreciate the opportunity to put a name to some morphologically similar species, but do you worry about the loss of traditional taxonomic analyses? Many universities are losing their organismal biology departments to DNA workers.
I agree with Matt. It is a worry. Personally, to me, where I am, it's less of a worry than it probably once was, because in this country, maybe 10 years ago and as recent as 5 years ago, barcoding was sold as the answer to everything, and you'd be able to very, very readily get a species ID by a little handheld kit that every naturalist would be carrying around in their back pocket. I still think, well, that technology sort of exists, but I think we're still a long way from that position. And I think there's always going to need to be taxonomists around to put into context what the DNA is saying. I'll give you a very brief example. Again, back to the species from feces nature metrics work. The two craneflies that were identified, as I said previously, one of them was associated with sphagnum and bog habitats. And as soon as I saw the list, it rang an alarm bell, because I thought that there's no suitable habitat within Buckinghamshire, or certainly Buckinghamshire, of that type. So is it a mistake? Is the DNA barcode accurate for that species? Was it really lucky that this species that's new to a county just happened to be intercepted by a bat on that particular night and it ate it? Who knows? My guess is we probably have to question the DNA on that case. You know, do we have enough samples that specifically enable us to say that is definitely that species? So I think we're reliant on people with entomological expertise to work alongside our colleagues to put some sort of QA through this process, and so that we part govern the process. So it is a worry, but I think they leave us behind at their peril. I think there'll always be a place for invertebrate collections.
Yeah, I completely agree, Pete. And but I think this country, the United Kingdom, is a slightly different to the US in terms of a very long tradition of amateur entomology and a huge number of people doing it. You know, there's not so many professional entomologists, taxonomists, is there, in this country as it is? There's not many. Most, you know, some of the best taxonomists in this country are just amateurs, you know, and so they're not going to ever sort of lose their jobs or interest because they're doing it for the love of it. I think DNA will enhance if anything, the possibilities and study of natural history, understanding behaviours and all kinds of things. I think, but yeah, it's a topic that is very slowly unfolding. It's not just hasn't happened at the speed that might have been predicted. So let's see. Okay, well, I think we'll leave it there for tonight. But thank you so much, Pete. It's been a really excellent talk and really good set of questions, really interesting questions from the audience. And thanks everyone for coming. And I hope that you're able to, well, I guess many of you will already have the British Craneflies book, but I hope many of you are also able to attend one of these forthcoming workshops in the next couple of years and further your knowledge as well.